Welcome. The topic of this lecture is immunity and the immune system. We're going to take a look at the difference between active and passive immune responses and then clarify the difference between innate and adaptive or acquired immunity because those are key concepts. We'll also look at the concepts that are important in adaptive immunity, the antigen recognition, antibody production, how we use antigen-presenting cells, and MHC, or major histocompatibility complex. And then we're going to talk back and forth about the role, the maturation, and the activation of T cells and B cells and how they actually function in immunity. And then we'll end our conversation with describing the age-related transitions in immunity that you should be aware of and have a good handle on. So let's start by talking about immunity in general and make sure that we're all on the same page. We know that we have both innate or natural immunity and adaptive or acquired immunity. Innate and natural immunity begins with cellular injury. As a result, then, it depends heavily on natural barriers, our natural barriers like our skin, our epithelium, and depends heavily on the inflammatory response. If you've already listened to the lecture on inflammation, we talked about that in that huge process that is a part of our innate natural immunity. But we also have adaptive or acquired immunity. Adaptive or acquired immunity occurs after innate immunity has already been activated. And because you've already done some activation of your immune system through that primarily inflammatory response, we know that adaptive or acquired immunity depends heavily on T and B cells. Now, adaptive immunity comes in both active as well as passive processes. Our acquired or active immunity means that immunity was produced after exposure to an antigen or through immunization. Now, immunization also does provide some antigen, but the idea of the active immunity is the host organism had to specifically do something. Their immune system actually had to be exposed to an antigen, and then it triggered and produced protection. As a result, then, because the organism had to do something to acquire this immunity, it becomes long-term. Passive immunity does not require the host immune system to respond. It is immunity that is provided, and since it was given to the organism, it is temporary rather than long-term. So it's important that you be able to differentiate between the two. A good example of passive immunity is if an individual has had an exposure to an infectious organism like hepatitis and is given IgG. So they're given antibody, but they didn't have to produce antibody to the hepatitis. They were given the IgG. Passive immunity is also what infants depend on right for their first 6 months of life, having passive immunity from their mother. When we think about adaptive immunity, the key to adaptive immunity is the ability to recognize antigens and develop that long-term protection to a specific antigen. So again, this is a major difference between that innate immunity of inflammation and adaptive immunity, which is very antigen specific. Adaptive immunity requires antibodies, immunoglobulins, as well as lymphocytes, the lymphocytes being T cells and B cells. Recognition of antigen by B cells occurs in the thymus. By B cells, this occurs in the bone marrow. When lymphocytes pass through either the thymus or the bone marrow, they have the opportunity to mature, and at that point they mature into either T cells or B cells. Lymphocytes are not active until the antigen is present. So that's, again, very, very important, antigen specificity. As we look further in an introductory sort of way at T cells and B cells, we frequently think about T cells being primarily responsible for cellular immunity. and B cells being primarily responsible for humoral immunity. Now, they both cooperate in our adaptive immunity, but they do have different responsibilities. Let's take that then to patterns of pathology. So when we think about an altered immune response, a decreased immune response, if T cells aren't available or they're unable to respond for cellular immunity, what you'll see is in altered cellular immunity is a pattern of opportunistic infections, so frequent opportunistic infections. Great example of that is an individual who is HIV positive or who has AIDS and develops the opportunistic infection of pneumocystis jirovecii. It's an opportunistic infection because their T cells they have insufficient T cell response. If we think about an altered humoral response, we think about a pattern of systemic infection, especially bacteria that has a capsule around it. So very different responses, aren't they? You may also see a poor humoral response or a bad humoral immunity response manifesting as a decreased or a poor response to vaccines that are given. Because vaccines, you want humoral immunity. Now with recognition and response, we're going to go through these four major areas, and it's important, this is sort of definitional, I'm sorry about that, but it helps us with our nomenclature and then we can use it and then we know what they do. So it's important that you have a good understanding of these. So first we'll start with antigens. We've talked a lot about antigens, both on the inflammatory side, but also, we're going to continue talking about them when we talk about the immune response. So when you think about an antigen is something that stimulates the immune response. So an antigen has immunogenicity. It induces that immune response to produce antibody. So an antigen either reacts with an antibody or an antigen or antigen receptors or with T or B cells. An allergen is an antigen. A hapten is also an antigen. A hapten is actually a small molecule that by itself is not an antigen. However, when this small molecule that is a hapten, when it combines with something that is larger, it becomes antigenic. Antibodies are molecules that recognize antigens, very simply. And we know that antibodies include those classes of immunoglobulins. And these are the immunoglobulins that we think about all the time, IgG, A, M, and E. IgD, or immunoglobulin G, is also present, but it isn't as well known or well understood as G, A, M, and E. I guess I could have done that alphabetically, but I didn't. So what's an antigen-presenting cell? Antigen-presenting cells process and present antigen to those T cell receptors. So they become very, very important in our immune system. Macrophages are APCs, antigen-presenting cells. And then last but not least is our genetics, our major histocompatibility complex. Our major histocompatibility complex is a large group of very complex genes, tend to be on chromosome 6, and that's our genetic makeup as it pertains to our immune response. The MHC, or our histocompatibility complex, plays a very important role in human leukocyte antigens, HLA system, and that makes a lot of difference and matters in our transplant donor recipient decisions. These complexes, these HLAs, are glycoproteins that are on the surface of all of our cells except red blood cells. And then the function of our HLA, of our major histocompatibility complex, is antigen presentation. Let's spend some time then talking about T cells and B cells. We're going to move back and forth between B cells and T cells for the next several slides. First off, remember, every T cell and every B cell is antigen specific. So B cells and T cells come from common precursor cells, and these precursor cells are in the thymus or in the bone marrow. And these precursor cells are help us develop immune competent T or B cells. These immune competent cells then migrate to secondary lymphoid cells, and they sit there and they wait for an antigen. When an antigen shows up for them to interact with, that's why they then become antigen specific. So let's talk then about T cell maturation and then and then we'll also talk about B cell maturation. T cell maturation occurs primarily in the thymus. And these receptors, T cell receptors, are produced by multiple gene combinations. There are 2 chains and there are 2 primary chromosomes. When those chains join, they make an antigen-specific T cell receptor. As that T cell differentiates in the thymus, it develops CD2 on the cell surface, and then that serves as a marker for those T cells. With further development, CD4 and CD8 develops on the surface of the T cells. The end result then is 2 different groups of cells. You have CD4 cells and CD8 cells. CD4 cells become helper cells or T helper cells. CD8 cells become killer cells. So let's look at CD4 cells a little bit more closely because they become the helper cells. CD4 cells recognize antigen, and those CD4 cells then trigger B cells and T cells for the immune function. If you don't have CD4, you don't trigger that response. So it's the way those CD4 cells work, those T cells work. They have to be able to trigger that response because T cells don't produce antibody. They have memory, but they don't produce antibody. B cells develop in the bone marrow in a process that's kind of like the T cells. B cell receptors develop and have the ability then to produce antibody on the plasma membrane. As these B cells mature and develop, they actually have little tails that insert into the plasma membrane. B cells express IgM. They also develop surface molecules that are either complement or adhesion molecules. Now, the key to B cells then is the production of antibody. As an antibody titer increases, which means as the amount of antibody increases, the affinity for that specific antigen then also increases. As a result then, the immune response is more effective. How then do we process antigens? Remember, once again, T cells and B cells don't react directly with antigens. They do other things. Antigens are processed and then presented by antigen-presenting cells, like macrophages. Phagocytic cells process those antigens. They put them on the they then, after they process the antigen, they put them on those processed cells are on the phagocyte surface, where lymphocytes then initiate the immune response against the specific antigen. That's why we call them presenting cells. The primary cells that are involved include the dendritic cells, the macrophages, the B lymphocytes. Each one of these has a talent for varying effectiveness. So for example, macrophages are very good at presenting antigen to memory T helper cells for rapid response. B cells coat the antigen with antibody, and then the neutrophils phagocytize that antigen. As I said earlier, T helper cells, T helper lymphocytes, are very important in both our cellular and our immune responses, because remember, those T helper cells stimulate those T and B cells. It helps with the maturation of both the cells in an antigen-driven process of adaptive immunity. T helper cells have the ability to interact directly with the antigen-presenting cells, to differentiate and activate cytokine genes, to interact with immune-competent T and B cells, and as a result then the result is differentiation into either plasma cells, which would be the B cells, or T cells. So those T helper cells, those are very, very important in triggering and assisting the immune response. B cells, again, are very important in our humoral immune response. B cells become plasma cells after they encounter that antigen and differentiate with specificity to that specific antigen. As a result, then, you produce antibodies to that specific antigen that then started that differentiation process. Now, what's interesting in our humoral immune response, that B cell based response, is that there is both a primary and a secondary response. In the primary response, this is the first exposure. With the first exposure to the antigen, there's a production of IgM, and this occurs usually in about 7 days, and that then leads to the production of IgG. Because this was a first response, and so as a result, you would say that the organism was naive to that antigen, the levels will gradually begin to decrease unless it's continuously stimulated. So those levels will decrease. So then you come back with the second time that same antigen comes. So with that secondary response, this is what happens with second exposure. Because you have IgG, you have memory. Antibody is produced more quickly and in larger amounts than with that primary or that first exposure response. And IgG is especially the type of antibody that would be produced. As a result of the secondary exposure, you tend to have higher levels that then remain. So those levels will remain longer. But the key also to that secondary response is because you had the first response and produced memory, the second time that antigen comes back to the organism, the response is larger and faster. Great example of this is a drug allergy. So you've given a drug. Let's take a sulfa-based drug for urinary tract infection, because we know that sulfa is fairly allergenic. So you've given a sulfa-based drug. The first time the individual took that drug, they may have undergone that first exposure, that primary response. Didn't have any problems with it. They were on it short term, and so they didn't have any difficulty with it. However, a couple years later, 6 months later, whatever, they have another urinary tract infection. You once again prescribe a sulfa-based drug. When you gave it now the second time, they already have memory to that antigen that they didn't like, right? They produced antibody. So when you give that sulfa-based drug, that allergen the second time, the antibody is there. You get a faster response to that. And so as a result then, the first time they didn't have any trouble with it, the second time they get a drug rash. Great example of primary and secondary response. Also works with peanut allergies, things of that nature. T cell activation is part of our cellular immune response. Like the B cells, the T cells are activated by antigen that binds to receptors, proliferates, and differentiates in response to the antigen. Now, activated T cells, remember, they don't produce antibody. However, activated T cells either kill the antigen directly, or they function as T helper cells to aid and activate those other cells that we talked about. T T memory cells are also produced for a secondary immune response, which is why you'll get that secondary response. So they have memory, but they don't produce antibody. Let's talk then about some of the characteristics of antibodies. Circulating antibodies are responsible for our host protection, especially from infection. Protection can be our host protection, and the way these antibodies work can be through neutralization, which means they basically inactivate the antigen. They may work through agglutination, which means they make them clot and stick together, so therefore they can't function. Or they may actually cause cellular contents to precipitate out, so the cells don't work. Antibody titers measure the level of circulating antibodies, which is why after you give an immunization and you want to know whether the individual responded to that immunization, you draw a titer. So attenuated vaccines, you're giving an inactivated antigen, and those attenuated vaccines respond through neutralization. So when you have that immune response, they will neutralize. Antibodies, however, can also be stimulated by presenting them with chemically inactivated antigen, like a toxoid. This is what tetanus toxoid does. Now, antibodies also have the ability to function as opsonins. Now, remember that process of opsonization in inflammation? Opsonization is the ability to recognize and adhere to the antigen. As a result, then, that makes the antigen more likely to be phagocytized, because you have to have that recognition and adhering to the surface of the antigen. The other thing we know about the immune response and the ability to develop that especially humoral response is that our immune response varies throughout the day. And we've identified that response to vaccine then is higher in the morning than it is later in the day. One of the more recent studies that was done looked at whether or not an antibody response to, I think it was the flu vaccine, was better in the morning or later in the afternoon in a group of individuals over the age of 65. And this was done at the University of Alabama, and they looked at about 276 individuals. And they randomly assigned the 276 patients to receive the vaccine either in the morning between 9 and 11 or in the afternoon between 3 and 5 p.m. Response to the vaccination was higher, significantly higher, in the group of individuals over 65 years that received their vaccination in the morning as opposed to those who received their vaccination later in the day. So recent research. We would not have a complete conversation or discussion of the immune system if we didn't at least recognize the importance of the secretory immune response. The secretory immune response is also part of our systemic immune system, and this is our mucosal system and our secretory organ system. Plasma cells in our secretory organs produce antibodies and secrete these antibodies in their secretions. So when we think about those secretory organs that have the ability to place antibody in their secretions, you think about our tear ducts, our salivary glands, the breasts. The breast tissue is a secretory organ. The bronchi, the GI, and the GU tract, mucous membranes. Specifically, what we look at are immunoglobulins, and these immunoglobulins are secreted locally as well as act locally. IgA is the most common of the immunoglobulins that we frequently think about, and generally that's in relationship to breast milk. So let's talk a little bit about at the end of our discussion about how we change as we get older. And we're going to look at our transitions and our immune system early in our age and later in our age ranges. So let's first think about our transition from the fetal to newborn life. When we are born, we have an immature immune system. Our leukocytes are not only less lethal, they are less effective as phagocytes. So our leukocytes don't have that killer instinct yet. They they are frequently slower because they haven't had a chance to develop, and they aren't as hungry, so they aren't as effective. Right around 32 weeks of gestation, or at 32 weeks gestation, maternal antibodies move through the placenta to the fetus. So this would represent passive immunity. Passive immunity when mom passes the antibodies to the continued developing fetus, lasts about 6 months after birth. It may last as many as 10 months, but most of us would say about 6 months. So this would be very, very important if you work with premature children. So those of you who are in the neonatal nurse practitioner program, this has a lot of implications for you pathophysiologically, as well as all of you in the FNP and the pediatric program, because you're working with newborns. So remember that this is passive, therefore temporary immunity that, if we're lucky, will last about 6 months, because those maternal antibodies, the antibodies were delivered to the baby. The baby didn't produce their own antibodies. We know that IgA especially, but also IgG and IgM is secreted in breast milk. So this then goes to the immune function of the secretory organ of the breasts. Data indicates that in infants that are breastfed, there's a decrease in otitis media, upper respiratory infection, and GI infection during early infancy. Now, in the meantime, during this 6 months of passive immunity, the newborn is producing their own T and B cells, but again, it takes about 6 to 8 months. Now, if you look at an immunization schedule then, and you start looking at when we start giving immunizations, you can see how we're taking advantage of the developing newborn's immune system. The job of this course isn't to teach you those immune schedules, but believe me, you're going to learn them through your program. At the other age range, we need to look at our transition as we get older. And when we're looking at transition as we age, as much as I hate to admit it, this does start a little bit in our 60s, and certainly in our 70s, 80s, 90s, and 100 plus years. We know that as we get older and older, we produce fewer T cells. So we have minimal production of new T cells. And we have evidence of that, and we know this because if you look at the thymus gland, as we get older, the thymus gland becomes smaller. And remember, that's where we were maturing and producing them. We also know that B cells are less responsive to new antigen, as well as having a poorer recall to previously recognized antigens. So what this basically means is, as we get older, our T cells don't really care if they are less caring about meeting new antigens. But more importantly, they're more forgetful. They've forgotten that they have already met this antigen, and so they don't respond as effectively. And then in the inflammation lecture, we did talk about the concept of inflammaging, that link between chronic illness and chronic inflammation. And we know that chronic illness is almost across the board is more likely to occur as we get older as opposed to our younger years. So let's see if we can pull a few things together. We know that immunity is absolutely essential to homeostasis, and it gives us both our first and our second line defense. Our first line defense depends, remember, heavily on inflammatory response, and our acquired immunity depends, yes, on our T and B cell response. So we know that we need both of those pieces. We know that in order to develop specific and lasting response to an antigen, we have to have adaptive immunity stimulated. And that the key to that adaptive immunity is the T cell and the B cell function, both of which have specificity to antigen, both of which have memory, but only B cells produce antibody. With humoral immunity, the first response to the antigen is largely quiet and results in memory. The second response is rapid and then long-standing. Remember that with T cell and the focus on cellular immunity, if someone has a deficiency, you're going to see a pattern of opportunistic infection. If someone has a deficiency in their B cell function, their humoral immunity, you're more likely to see a pattern of systemic infection, especially with bacteria that are encapsulated. So I think that probably gave you a couple of hints of things upon which you should focus. I appreciate your attention, and please let me know if you have any questions as you review this information.